Welcome to the Deep Dive. Today we are doing something a little bit different. As an expert in two-photon polymerization, you're probably used to seeing your field leveraged for things like incredibly intricate micro-optics.
Right, or photonic crystals.
Yeah, exactly. Or, those famous microscopic 3D-printed boats floating around in a single drop of water.
I mean, those are great for demonstrating the tech, but today we are completely pulling 2pp out of the clean room.
We're applying it directly to the human body. We're looking at a fascinating paper from a 2025 issue of ACS Materials Letters. It details something called the allergy patch.
And this is where it gets really relevant for you listening. The researchers here utilized 2pp to engineer a wearable microneedle-based electrochemical biosensor.
Which is designed to continuously and critically non-invasively detect food allergy sensitization.
Yeah, this paper brilliantly bridges high-resolution nanofabrication with critical biomedical diagnostics. Because we often see 2pp relegated to in vitro lab-on-a-chip applications.
Right, mostly because of the fragility of the structures, right?
Exactly. Taking the precise structural mechanics that only two-photon lithography can provide, and then applying them to a harsh, dynamic biological environment.
Like human skin.
Yeah, like human skin. It represents a massive leap forward in continuous patient monitoring.
So to understand why this specific geometry and, high-resolution fabrication were even necessary, we have to look at the clinical target first.
We are talking about food allergies.
Right, which, if you or anyone close to you suffers from them, is just a constant, terrifying reality. The ultimate fear is anaphylaxis.
That severe, potentially fatal systemic reaction.
Where airways swell up, blood pressure drops rapidly. And the biological culprit driving this entire cascade is an antibody called immunoglobulin E.
Or IgE for short.
Right. When a sensitized individual is exposed to an allergen, that IgE cross-links with receptors on mast cells, and that triggers a massive release of histamine.
And the clinical bottleneck we're currently facing is how we actually detect those dangerous IgE levels before that extreme reaction occurs.
Especially in vulnerable populations.
Exactly. The standard testing methodologies are just fundamentally flawed for continuous monitoring. I mean, skin prick tests literally involve exposing the patient to the allergen.
Which seems wild when you think about it.
It is. You're observing the localized inflammatory response, but in highly sensitized individuals, the test itself can trigger systemic anaphylaxis.
Making the diagnostic tool itself potentially dangerous.
Right.
And the alternative is blood tests, looking for a specific IgEs or total IgE levels. But blood draws are invasive, they're painful, and they suffer from massive patient compliance issues.
Particularly in pediatric care.
Oh, absolutely. Trying to get a toddler to sit perfectly still for a venous blood draw, just to figure out their baseline sensitivity to peanuts or dairy, it's a nightmare for clinicians and parents alike.
And beyond those logistical hurdles, the target concentrations require highly sophisticated detection methods.
Give us the numbers on that.
So in an adult who is not susceptible to allergic reactions, normal circulating IgA levels hover around 150 nanograms per milliliter.
Okay, 150.
But in sensitized adults, that concentration generally exceeds 240 nanograms per milliliter.
Right, and just to put that in clinical terms, 1 international unit equals 2.4 nanograms.
Exactly. But you have to remember, IgE accounts for less than 0.01% of the total immunoglobulins in the human body.
So we are essentially looking for a microscopic needle in a biological haystack here.
We are, and the challenge is even more pronounced in pediatric care because IgE levels naturally increase as a child ages and their immune system develops.
Meaning the threshold for a dangerous sensitization in young children is significantly lower than that 240 nanograms per milliliter mark.
Precisely. A viable diagnostic tool needs astronomical sensitivity to be clinically relevant across all age groups.
But here is the incredible part, and this is the biological loophole the researchers found that eliminates the need for a blood draw entirely.
The interstitial fluid.
Yes. IgE doesn't just circulate in the bloodstream. It also exists in the dermal interstitial fluid. That's the fluid bathing the cells just beneath the outermost layer of the skin.
And the crucial detail from the source material is that IgE is present in this fluid at a 1-to-1 ratio compared to blood serum.
A perfect 1-to-1 ratio. So if you can tap into that skin fluid without digging deep enough to hit the capillary beds and nerve endings further down,
you can accurately measure allergy sensitization without a single drop of blood.
Which brings us to microneedles. They are the obvious solution for accessing interstitial fluid.
However, Standard microneedle arrays have massive limitations when you try to adapt them for biosensing.
Right, because a traditional hollow microneedle is basically just a microscopic hypodermic syringe,
And it almost universally gets clogged with epidermal tissue or cellular debris the second it is inserted into the skin.
which completely isolates and ruins any sensor you've placed inside it.
Exactly. Now, the other common manufacturing route is making the microneedle entirely porous.
Like creating a microscopic sponge so the fluid can just passively diffuse inward.
But as a 2PP engineer listening to this, you already know the structural trade-off there.
Yeah.
High porosity inherently sacrifices mechanical strength.
Right. A purely porous microneedle simply snaps off.
It snaps off and remains embedded in the skin because the material just lacks the rigidity to penetrate the tough stratum corneum.
So the researchers realized they needed a geometry that traditional micromolding or drawing techniques simply could not achieve.
Which is exactly where your field provides the ultimate solution. They turn to nanoscribe 3D lithography.
To print a hybrid architecture, it is both hollow and porous at the same time.
They utilized IPS photoresin, which is a biocompatible carbamate methacrylate-based resin, to build this completely novel structure.
And looking at the fabrication specs, they really pushed the aspect ratios here.
They did.
Printing these structures requires serious optimization of the hatching and slicing parameters. The needles they fabricated are exactly 700 microns tall,
With a 300-micron base diameter.
tapering up with a wall thickness of just 50 microns.
And along that incredibly thin 50-micron wall, they printed uniformly distributed 40-micron wide pores.
Just incredible precision. And to maintain the collective structural integrity of the patch and allow for adequate fluid exchange, the tip-to-tip spacing between the needles on the array was set to exactly 500 microns.
That 500-micron spacing is a highly calculated parameter. It avoids the bed of nails effect.
Right, where if they're too close together, it just pushes the skin down instead of piercing it.
Exactly. The surface tension and elasticity of the skin distribute the applied pressure across multiple tips, preventing any single needle from actually piercing the stratum corneum.
By spacing them at 500 microns, they ensure each tip applies sufficient localized pressure to penetrate.
While still retaining enough density across the patch to draw a clinically relevant volume of fluid.
But the mechanical validation is everything when you are dealing with a wearable patch.
Yeah.
Nobody wants a microscopic lattice of polymerized resin breaking off inside their arm.
Absolutely not.
We have this 700 micron tall structure with 50 micron walls full of 40 micron holes. The challenge during the development phase alone, just washing out the unpolymerized resin without those walls collapsing under capillary forces, is significant.
So how did this hybrid hollow porous structure actually hold up against the dense layers of human skin?
The researchers put the arrays through rigorous mechanical compression tests using a universal testing machine.
And they determined the fracture force of the 2pp array. The sheer mechanical load required to crush the printed structure was 1.85 newtons.
Okay, 1.85 newtons to crush it.
Right. Then they measured the piercing force, which is the actual load required to successfully puncture an artificial skin model.
They used a dense agarose gel for that, right, to mimic human tissue.
Yes, and they subsequently verified the results on ex vivo human abdominal skin. The piercing force was only 0.27 newtons.
So the 2PP technology allowed these engineers to create a device with a greater than six-fold safety margin.
Exactly. The force required to catastrophically fail the needle is more than 6 times higher than the force required to successfully insert it.
That hybrid structure is rigid enough to pierce the skin, but porous enough to let the interstitial fluid flow in without clogging the inner chamber.
Creating the physical structure is a monumental achievement, but it really is only half the battle. They had a mechanically robust, biocompatible fluid extraction system.
The next step was functionalizing that highly engineered resin into an electrochemical biosensor.
Right. IgG is the ultimate stress test for this device.
Structurally, IgG is incredibly similar to our target, IgE.
And more importantly, in human blood and interstitial fluid, IgG is roughly 20,000 times more abundant than IgE.
The sensor has to find one specific molecule in a sea of 20,000 near-identical decoys.
But when they ran the interference matrix, the patch produced a signal that was 10 times stronger for the target IgE than it was for the overwhelmingly abundant IgG.
That level of selectivity perfectly validates the intricate design of the D17.4 AB tamer and the stability provided by the shielded 2PP architecture.
And to ensure these mechanics translated from pristine buffer solutions to complex biological tissues, the team advanced to ex vivo human skin trials.
They utilized excised human abdominal skin, pre-incubated with varying concentrations of IgE to simulate a highly sensitized patient, and then applied the microneedle array.
Moving from agarose gel and buffer solutions to actual human tissue introduces a massive variable in fluid dynamics and tissue elasticity.
But running the square wave voltammetry at a 300 Hz frequency, the patch performed exceptionally well.
At a concentration of 1 microgram per milliliter of IgE, the researchers recorded a massive 500% signal enhancement.
It delivered a clear, dose-dependent electrochemical spike that perfectly correlated with the IgE concentration hidden within the dense dermal layers of the ex vivo tissue.
This deep dive fundamentally demonstrates a shift in how we apply two-photon lithography. We are moving past microscopic art and lab-on-a-chip novelties.
The 2PP process enabled the exact structural mechanics, the hybrid hollow-porous geometry of the 40-micron pores, the 50-micron walls required to bring continuous, painless, real-time immune system monitoring out of the lab and into a wearable clinical device.
We are looking at a technology that could eventually end the reliance on pediatric blood draws for allergy testing entirely.
Which is life-changing.
It is. The Allergy patch proves that 2PP can create intricate, shielded architectures capable of protecting delicate biochemical sensors from the intense physical friction of penetrating the human body.
And as an expert in 2PP listening to this, consider the broader implications of this design philosophy.
What other harsh, high-friction biological environments could be unlocked by this approach?
Exactly.
Could we 3D print shielded, porous microstructures that protect active surfaces on the inside to navigate the highly acidic and mechanically abrasive environments deep within the digestive tract?
Or perhaps deploy them within the high shear environments of our cardiovascular systems to monitor localized biomarkers.
The precedent has been set here for turning structural protection inward.
The potential applications expand exponentially when you rethink where the active surface of a sensor should actually live. Thank you for joining us on this deep dive. We hope this look at the Allergy patch and the evolving role of two-photon polymerization inspires you to keep pushing the boundaries of nanofabrication. Keep innovating, and we will catch you next time.